Welcome to the AP Bio podcast for Chapter 10, Mendel and Genetics. What is genetics? Genetics is the study of heredity, which is a property of life. Heredity is the transmission of traits from parent to offspring. Who is the father of genetics? That is Gregor Mendel. Why was Mendel successful as the father of genetics? There were other scientists doing the same experiments at the same time, but why was Mendel successful? There are two reasons. Number one, he used a garden pea as his test subject. There are 6 reasons he chose the pea. Reason number 1, they were small, they were easy to grow, they produced many offspring. From one cross he would get hundreds or thousands of data. Mature quickly, there's many varieties of the pea, and they're easy to fertilize. Peas can self-fertilize, which means they fertilize themselves because they have both male and female parts on them, or they can cross-fertilize. One plant can fertilize another. Here are the different varieties of the pea. See, when you think of a pea, you probably think of the green pea, but there are also yellow peas, round peas, wrinkled peas on their pod, inflated versus restricted, green or yellow, the way they grow on their stem. The second reason why Mendel was successful was that he used a quantitative approach, which means he counted his results. This was Mendel's experiment. This is what it looked like. Step number 1, he needed to produce a parent generation. We're going to call this generation the pea generation. He allowed pea plants to self-fertilize for many generations. This made sure that he had pure parents that were true breeding or pure. For example, he used one pure purple pea plant and he used one pure white pea plant for his pea generation. Step number 2, he needed to produce the first generation, or the F1 generation. F stands for filial. So what he did is he took his 2 pure pea generation pea plants and he cross-fertilized them. What was his result? Instead of getting some purple and some white, he got all purple. 100% of his peas were purple. None of them were white. So he saw, what the heck? Why weren't any of them white? So then he went to step 3 and he produced the second generation, or F2. He took 2 of the F1 generation pea plants and he self-fertilized them. What were the results for this one? 75% of his plants were purple. 25% of his plants were white. And this repeated no matter what type of trait he was looking at, it always got a 3-to-1 ratio. Here's a picture diagram of his results. For his pea generation, he had a pure purple and a pure white plant. He then cross-fertilized them. Then he got all purple for his F1 generation results. He then self-fertilized that and got a 3-to-1 ratio of 3 purple to 1 white. So what did he conclude? He concluded heredity. He figured out what heredity was. Parents can transmit information about their traits to their offspring. He figured out that each individual pea plant, or now we can relate this to humans, have 2 factors for each trait. He didn't know what a gene was, but that's what he was referring to. Every individual has 2 genes for each trait, one from mom and one from dad. These genes or factors are represented by letters, and these letters are called alleles. Alleles occupy corresponding loci or locus on homologous chromosomes, as we saw in the last chapter. All right, an allele. If both of the alleles that you get from mom and dad are the same, the individual is homozygous for that trait. If both alleles are different, you are heterozygous for the trait. Traits can be described in 2 different ways. When we do genetic problems, I will be asking you for a genotype. Genotype are the letters or the alleles that represent the trait. For example, there are 3 genotypes possible for this trait: big P, big P, big P, little p, and little p, little p. When you are writing out genotypes, make sure you can tell the difference between the capital and the lowercase letters. And that is what it looks like. What is the expression? For example, purple and white. Those are phenotypes. More about traits. Only some traits can be seen. Others are masked. Dominant traits. Dominant traits only need 1 capital letter to be expressed. Recessive traits need both letters, or it's masked, so you can't see it. And those are represented by the lowercase letters. All right, so let's see if you understand. So, for big P, we're going to do big P, little p, and little p, little p. For big P, it's a dominant or recessive. It is dominant because it has at least 1 capital letter that makes it dominant. Is it homozygous or heterozygous trait? It is homozygous because both of those letters are the same. And so what would be the phenotype for our P example? That big P would be purple because purple was dominant in Mendel's experiment. Next genotype is big P, little p. That is dominant because of the capital P. So even though it has a lowercase p, it is still dominant. It is heterozygous because those letters are different. And it will still represent purple because of the capital letter. Little p is recessive. You need 2 lowercase letters to be recessive. It is homozygous because those letters are the same. And because it's recessive, that pea plant would have been white. We talked about locus. We did this in the last chapter. Remember on homologous chromosomes, one from mom, one from dad, for example. This is the spot where the pea gene is at, and those are called loci, or locus for individual. Little a, big B, little b. So mom and dad's genes are in the same location. Mendel came up with two laws. Mendel's law of segregation. This is what it states. In order for sexual reproduction to occur, the two alleles carried by an individual parent must become separated. If we have a plant in the F1 generation that is big R, little r, when that fertilizes and we want to get the F2 generation, we separate those letters. So big R is going to go here into one sperm, and the little r would go in a different sperm. Or the big R would go in one egg or ovule, and the little r would go into another. They are separate. As a result, each sex cell formed contains one allele of each pair. So, for example, the probability of the egg being little p is 1/2, right? Because you're either big P or little p. And the probability of the pollen being little p is also 1/2. So the probability of the offspring being little p is 1/4. You would multiply those together. This is called the rule of multiplication. The probability of getting a little p pollen and a big P egg is 1/4. That's what we just did. 1/2 times 1/2. The probability of getting big P pollen and little p egg is 1/4 as well. 1/2 times 1/2. So the probability of getting an offspring who is big P, little p is 1/2. 1/4 plus 1/4. This is called the rule of addition. Mendel's second law is the principle of independent assortment, and this states that members of any gene pair segregate from one another independently of the members of other gene pairs. For example, if you have big P, little p, if you have big P, little p, and let's do another trait, big T, little t. So we're talking about two traits, purple and tallness, okay? What this is saying is that the members of the gene pair segregate from one another independently of the members of the other gene pairs. So when we're forming our sperm, we got 4 sperm being formed from meiosis, what this law is saying is that this sperm isn't going to get the big P and the little t and be the super sperm. And this sperm isn't going to be the little p and the little t and be the mentally challenged sperm. It is random. It is random of which ones are going to go to which sperm. They assort independently of one another. All right, monohybrid crosses we will cover in class, but I'll do this really quick. Monohybrid crosses involve one trait. They cross 2 alleles on the same locus. They use a 4-box Punnett square. So if you were to cross a white flowered pea plant with a heterozygous purple flowered pea plant, those are your genotypes of your parents. You would put your little letters on the outside of the Punnett square, put your other parent on the outside of the Punnett square, and then you would bring the letters into each of the boxes. And then I would ask you for genotype percent, which is 50% big P, little p, and 50% little p, out of all 4 boxes. Phenotype percent would be 50% purple right here, and 50% white. How do you find out whether an individual is big B, or big B, little b? You know they're dominant, but you don't know what genotype they are. Scientists can do a test cross. A test cross is a technique that takes the unknown genotype and crosses it with the recessive individual. Once you do that, you can look at the results and figure out what it is. Another cross that we're going to do in class is dihybrid crosses. They involve 2 traits. They cross individuals with 4 alleles at 2 different loci. They use a 16-box Punnett square. When both genotypes for both individuals are heterozygous, the phenotype percentage will always come out to be 9:3 to 3:1. For example, across a homozygous purple flowered heterozygous green pod pea plant with a white flowered yellow pod pea plant. And we're going to say green is dominant over yellow. So here is our genotypes from the problem. In order to get the cross, you have to do FOIL to get the gametes, which is first, outer, inner, last. So for big P, big G, little g, F is going to be big P, big G. O is big P, little g, outer. I is inner, big P, big G. And L are the last ones, big P, little g. FOIL for little p, little g are just all the little letters because there's no mix there. Then these are the letters that you would put outside of your box. The answers to your FOIL go on the outside of the box, and then you just bring the letters down the same way you did before. So because these are repeating, this repeats down, and you are allowed to do little repeat marks or arrows going down. So then once you get your results, you would do your genotype percents and your phenotype percents, just looking at all 16 boxes and putting them into a percent or a ratio. Dominant recessive relationships. There are diseases where the homozygous recessive organisms would not survive. These are called lethal recessive, and there's examples. There's also incomplete dominance, codominance. For incomplete dominance, the heterozygote is an intermediate between phenotypes of two homozygotes. Blending will occur. So there isn't quite a dominant trait. There's a blend. This occurs in Japanese 4-o'clock plants or snapdragons. For this, you need to memorize that big R is going to represent red plants. WW is going to represent white plants. And so if you get a blend of that, if you give one of each, you get a pink plant. There are no lowercase letters here. So cross a red flowered Japanese 4-o'clock plant with a white flowered 4-o'clock plant. It would be RR with WW. Put your letters on the outside like you would do either way. And your boxes would be all RW. Usually you would write the R first. So the genotype percent is 100% RW and the phenotype percent is 100% pink. Okay, another one is codominance. This is when two traits share dominance, like in human blood types. There are special notations as well when doing these problems. IA, and IA little I is how you represent blood type A. IB, and IB little I is how you represent blood type B. Blood type AB, which is a cross mix of those, is IAIB. That's the only genotype for that. And blood type O is recessive, and that's little I. So cross a person with blood type AB with a person with blood type O, you would have IAIB times little I. And that's how you would set it up, just like your other crosses. Always write your dominant letters first. So IA little I, IB little I, IA little I, IB little I, and then you have your genotype and phenotype. The last one is sex-linked genes. These are genes that follow the transmission of the X chromosome. It's always expressed in males because they only have one X, and it's considered to be dominant. Females may express it depending on what they get. Hemophilia is an example, and that's what we're going to use for our problems. So, notations for sex-linked problems for hemophilia. Females first. X big H is a normal female. X big H, X little h is a carrier, or that is also called heterozygote. X little h is a girl who has hemophilia. For males, X big HY is a normal male. X little HY has hemophilia, and that's all you get. You either have it or you don't for males. That's why males get it more than females for these types of traits. So I want to cross a hemophiliac man with a female carrier. My female carrier is X big H, X little h, and my hemophiliac man is X little HY. Make sure you're always crossing a girl with a boy. Put the letters in together, just like you normally would. Sorry, that H is supposed to be right there. And then you do your genotype, phenotypes, percents the same. For genotype percents, they're all different, so you would have to write it all out, 25%, and write out each one. And then phenotype percents, you would just do it basically, do they have hemophilia or they don't? And 50% of them are normal, right here, and 50% of them have hemophilia.